Welcome to MOOC course on Introduction to Proteogenomics. In last lecture, Dr. Prateek Jagtap talked to you about the workflow of RNA sequencing to FASTA database creation. In today's lecture, he is going to continue the discussion about bioinformatics solutions for big data analysis. He will talk more about the output files and data analysis to understand the unraveling the questions in hand. He will also be talking about 3 workflows: RNA sequencing to variant FASTA database searching using MS data, and identification and visualization of novel variants. He will explain about search GUI, a set of freely available algorithms for mapping the MS data to peptide sequences. He will also talk about Lorikeet viewer, a spectra viewer of all the B and Y ions identified in the mass spectrum. I hope it will also refresh you about previous lecture by Dr. Karl Klauser, where he gave you the understanding of manual interpretation of data sets. So, let us welcome Dr. Prateek Jagtap for his today's lecture.
So, these two tools, both of these workflows, this one as well as this one, generates a protein sequence FASTA file as well as a mapping file, right. So, this is a protein sequence FASTA file. As you can see, there is the ensemble identifier here along with the name of the protein, if it's if the function is known, and this is another one. And the genome mapping file basically gives you again the identifier of the or the accession number of the protein, which chromosome it comes from, what's the start site, what's the end site, and it also gives you the length of the exon. Now, one of the things you will observe here is this is basically saying that this is a 174 base pair sequence or basis sequence, and then you can see that there is some gap between this and this, which basically shows that there is an intron present there, right. So, this kind of helps you to map your regions of the protein. And later it actually also helps you because if you just identified a peptide from this protein, one can use these coordinates to go back and find out what are the coordinates of the peptide as well. Okay, so let's imagine that we have run that workflow, right? This workflow. We started with the FASTQ files and the FASTA file as well as the GTF file, ran through all of this, and you got a protein sequence file. And so, this is how your history looks like once you go through the documentation. Once you go and access the Galaxy instance, go through documentation. After the first workflow, you have this as a history. And one of these here would be a protein FASTA file that you can use here for your analysis. So, the second part here is you have your RNA-seq database or protein FASTA generated from your RNA-seq database. You have your MGF files which you have acquired from the same data, and now you search it with, we have got a tool called Search GUI, which basically has at least 9 different search algorithms in it. So, it helps you to identify peptide spectral matches, and then it uses peptide shaker to perform both FDR analysis as well as protein grouping. And then there is another tool called as MZ2 SQLite that is used to perform further analysis. And it also generates peptides so that you can perform BLAST-P analysis. And BLAST-P analysis is important because you want to go back to the NCBI-NR database and ensure that these peptides do not match to what you have. And there have been some instances wherein something that we found to be a novel peptide just a month before is no longer novel because somebody annotated it, right. So, you have to ensure and even report in your manuscript that December of 2018 this still was a novel peptide. Now, in January maybe it is not, but at least you have covered your requirements by saying this is the database I searched against on this date, right. So, I might actually skip this slide, but this is a really, I mean, I use this slide a lot to explain the complexity or the process of how we start with proteins, digest the peptides, put it through LCMS, LCMSMS, identify them. But this basically captures everything, and you have perhaps seen this many times, so I won't go through this. But this is a really good article. This comes from one of the from Jürgen Koch's lab in Germany, and it covers most of the contemporary proteomics research that's going on. So something I would suggest to go and read if you're really interested in basics or even contemporary status of proteomics. So again, you take your spectra, you match against your protein database, and you can identify your peptides. Search GUI, as I said, is a freely available software. You can also run it as a GUI, but we have got it in Galaxy, so it kind of runs in the workflow that I mentioned. And then there is Peptide Shaker, which basically does protein inference, does FDR analysis, and identifies not only peptides, PSMs, but also proteins from your sample. It also generates an MZ identimal file, which you can use it for further processing. So in this second workflow, right? So we are in the second workflow now. We have got our protein FASTA from RNA-seq data. We have our MGF files, and we want to search that. So what it does is it takes those two inputs, searches the data, uses peptide checker to generate multiple outputs, and then this goes into a tool what we call as MZ to SQLite tool. So what it does is it takes any tabular outputs and generates a SQLite database out of it. What it helps you to do is it helps you to perform some more complex analysis on your tabular inputs and get outputs from that. So instead of you processing these tabular files in multiple processes, you can have it in one place and then perform more complex queries on that to get answers, right? And eventually what comes out of this is you identify peptides that we think are novel, in the sense they are not present in your reference proteome, and then you can take those peptides and subject it to BLAST-P analysis. So that kind of covers these two workflows. The first one which generates a protein FASTA file, the second which gives you two outputs. One is your peptides that are going to be subjected to BLAST-P analysis, and it also gives you a MZ2 SQLite file, and I'll talk about it, what it means, right? So the third workflow, which I think is the most interesting, because you kind of, these two workflows you can in some way, maybe the second one you can use it, you can use any other software to do it, right? You have an MGF file, you have your RNA-seq generated protein FASTA file, you can use any search algorithm to get your outputs. But these two workflows are something that you can, with the work that we have done, hopefully helps you to get it done easier on your data sets. So the third workflow, so by the end of this, your history would look like this. It will have 54 items, and there are 54 of these because you've kind of added, outputs from each of these tools. Again, and this is just as a backup in case your search hasn't run because it got stalled or because of some reason, you can always go back to that Galaxy instance I talked about and download the history for the third workflow. So again, the same process. You take your active history, run it with the third workflow, and what does the third workflow have? The third workflow basically takes in inputs from workflow 2, which is this BLAST-P, peptides for BLAST-P. It also takes the PSM report, so the peptide spectral match report from Peptide Shaker, and this is generally at 1% global FTR. And then it also has this MZ2 SQLite file. So this is basically all the information in your MZ identimal file, right? The MZ identimal file is the file that is generated by a protein or PSM search. That particular file, all that MZ identimal file information gets into that. And this is really important because it also has information about continuous B ions, continuous Y ions, all the spectral annotation information, and that's something that you can use to process your data.
MGF file. So all the information is there. So why do we need a MG ident XML file?
Right. So MGF file basically is just your peak list, right? It doesn't have any peptide annotations, right? while the MZ identifier file has that and has got a lot more information.
If my software is not giving me MZ ident XML file, so from where I should convert that my raw data to MZ ident ML file?
I mean, if you want to use the the next tool, which is the ability to look at the spectra, right, view your spectra, then you should have a MZ identifiable output in general. And there are many software which I mean, Scaffold does that, Protein Pilot does that. I'm sure Proteome Discover does that, though I haven't used that. So there are, MZ identifiable is quite a standard output nowadays that most of the software generate. So, but that doesn't, I mean, so that will, if your software doesn't do that, then it will avoid you to do the spectral visualization, but that doesn't mean you cannot do the rest of the things in this workflow. But again, if you run it in Galaxy, since Search, Go and Peptide Checker do that, you can use that, right? And then we also had this genomic mapping file from workflow 1, right? So all of these are actually used in the last workflow, and what it does is, so this tool here, which is the query tabular tool. So remember I talked about taking tabular files and generating a SQLite database out of it. The SQLite database now can be queried, and you can only say show me those peptides or peptide spectral matches which are novel, right? So you're matching it against the known database and try to find that. And these novel peptides, these novel peptides would be a result of this NCBI blast that you've done, right? And these novel peptides or novel PSMs now can be converted to just peptides, and these peptides can be subjected to generating an output which can give you peptide, its genomic coordinates and other information. So I will basically talk a little bit about that. So there are two tools used here. One is peptide genome coordinate and peptide pointer. So looking into the details, right, we had this BLAST-P peptide from workflow 2, this also from workflow 2, and this one from workflow 1. And then, by NCBI BLAST-P, you use certain rules, and these are the rules that we used, is if the peptide, if its BLAST-P ident is less than 100, or there is at least one gap present, or if the blast length is lesser than your query length, then you basically call it a novel peptide. So, and we have kind of used this on multiple data sets, and these three features seem to be enough to identify a novel peptide. So that's the information used here to identify novel peptides. And then it uses this tool called as pep pointer to generate a tabular output that you can use for further analysis. But before that, I would like to maybe talk about this thing called as MZ2 SQLite, which is used as an input to visualize your data. And I would really if you're really interested in spectral visualization, I will strongly encourage you to go through that, the Galaxy instance and the documentation, because this really doesn't take time. Each workflow takes, I think, the first one takes 12 minutes, the other takes just 2 minutes each. But it will help you to go through that. So what the MZ2 SQLite data does is it takes this MZ identifiable file that you have and generates a list of all the peptides that have been identified with associated information. Now you can select novel peptides. So remember, we actually identified some novel peptides here, which are already in the history. You can use that. So you could say, just show me the subset of these novel peptides, right? And then if you do that, this list goes down, and then by using various tabs that are open here, you can visualize the spectra using Lorikeet. This is all within Galaxy. So you can use a Lorikeet visualization tool to look at which B and Y ions are, or which peaks are annotated with B and Y ions, how many of these are continuous B and Y ions, and so on. So basically look at the spectral quality. Because even if you have done 1% FDR, and if you're getting really great score for your peptide, it's not necessary that the spectral annotation is good. And I think it's important that if you're identifying a novel peptide, you at least have the means to to show or convince yourself and then the reviewer that this this is a peptide that indeed is novel and is definitely needs further interrogation. One can also perform genomic localization using that MVP tool that I mentioned. So again, in that tutorial there is this peptide that we looked at, and as you can see it's annotated quite well. Now the lorikeet viewer also is interactive in the sense you can check on B and Y ions and so on and so forth to select these. So this, if you were to do this manually, this would maybe take 20 minutes to half an hour for somebody to look at each of these, while this takes a few minutes or even a few seconds to go through this. It also gives you an option to say this is a good one or bad one, so that you can look at the list of all your novel peptides that you have here. Let's say if you have 200 of them, you can look at the spectral quality and then only select those that are important for you. The next part is genomic localization. So again, by using various buttons or various features, when you look at the protein view report in the MZ SQLite file, you can open the integrated genomics viewer and then you can look at what are the variations that you see, and there are you can lay down tracks and look at the RNA-seq data. You can look at the 3-frame translation of the DNA sequence, the annotated peptide sequences, and so on and so forth. So it kind of gives you a pretty good way of looking at the peptide that you identified, and you can also scroll and look at adjacent peptides and so on and so forth. So the eventual output that comes out of this is it tells you a chromosome number, what's the start and stop sign, what is the peptide that came out of it, and what's the annotation, where do these peptides lie. The ones that we found in CDS were basically the ones that were either single amino acid variants, or there were some that we found in the 5' untranslated region and so on and so forth. So if you have, let's say, hundreds of them, it helps you to identify or even classify these peptides based on the variants that you found. It also gives you something more interesting. You can sort this by chromosome, and then you might even see a pattern wherein these peptides maybe are lying around a particular region, and that kind of gives you an idea about mutation that could be occurring in that particular phenotype, right? You can use the information, the output that you generated from this. You can just take that link and open it in UCSC genome browser, and then you can look at more details about how was this gene or the homology of that particular gene in other organisms, and convince yourself that the mutation that you see is indeed novel or is similar to some other organisms, right? So again, there is information here that you can use it through UCSC browser, and in this case for that particular peptide I talked about, the same peptide, right? That's the localization we found, that there was a mutation in this particular region, that G had gotten converted into a. You can also, and this is not used in Galaxy, but you can also go beyond and use some other tools to look at which, what are the conserved domains that it occurs in. So I mean, basically, I would really strongly encourage you to go ahead and use that tool, because if you're interested in using proteogenomics or using Galaxy for proteogenomics, I'm sure most of you are interested in proteogenomics. And it kind of gives you a sense of how it can be used. Now it's again set up for a small data set, with something that works in maybe an hour or two. But you could also think about then upgrading that to using it for larger data set, your own data sets, for example. So I just wanted to mention that Galaxy P is not only about proteogenomics. We also work in the area of metaproteomics and metabolomics. So we are developing tools and workflows to enable that analysis as well. And then, so this is what I would encourage. Go try it out, look at this site. There are instructions. All you need is a registration and password, and a login and password. And if you use these documents, which are really detailed, and we have used it for at least 3 workshops this year, feel free to try it out. Again, these are the same documents. As I said, as a backup, if this fails, you can always try this instance. And as I was mentioning to somebody here, this would be a instance that would be there for longer. So if you want to just skip that and start using that, that's fine too. Just wanted to acknowledge the people who were responsible to make this happen. Praveen Kumar and Professor Tim Griffin. Professor Tim Griffin is the PI of the Galaxy grant, and Subina Mehta were the people who actually, so Praveen developed a few software along with James Johnson, but Subina Mehta actually tested them and also worked on the documentation that you can go and have a look at. Lastly, we work with multiple researchers around the world, not only users but also developers, because tools are getting developed as the field is emerging, not only in proteogenomics but other fields as well. We see that many tools are getting developed. So we try to work with the best tools that are available, try to package it in Galaxy and integrate into workflows. Links to you can actually go to galaxyp.org and find more information. There is also galaxyp.org/contact if you want to contact us. But with that, I think I'll be happy to take any questions.
Does the database incorporate the novel junction splice junctions which are there?
Yeah. So the mapping file that is generated from the second workflow would have that information.
But does your database currently have the list of novel identified splice junction sequences?
So yes, for a particular data set, it will have it, right? So you need to run your data set, your RNA-seq data to get that. So I mean, for this example, yes, we do have it. If you run some other data sets, which we have done, we have that. Yeah, so it is possible. So that's why we have those two different workflows. One which is single amino acid variants, which uses a different set of tools, and junctions, which is a larger genomic reorganization, right? So yeah. And it's not only captured in your genome mapping file, but it's also captured in a protein FASTA file. You'll have your genomic coordinates and the fact that it's a junction, a novel junction. I would encourage you to do a hands-on session later with the documentation, but I can show you what I was talking about. In the sense, I can show you this in action or where things are. And again, if there are more questions, I can take them. So this is the Galaxy instance, right? Does this work? No. So, for you to log in and register, this would be, so I'm already logged in into this, right? This is the tool format, and this is the history, and this is the pane, the central pane. So the way you start it is you go to shared data, and then there are histories. Click on history. So as you can see, there's an input history here, right? And if I click on that, it shows these are the 5 files or 10 files in fact that I have here, and I can explain why there are 10 files and not 5, and we can see only 6 of them, sorry. And then I go to this place called import history, and you can name this anything. So I can call this Mumbai, import it. Once I import it, it basically goes here. So this is just one way, this is for the tutorial, but you could also have all your data sets on your, or somewhere else, right, on a FTP site or on your computer, and you can upload that as well. So there's an upload function that's also available, but for the sake of this, so here as you can see, there's a FASTQ file, there's a GTF file, FASTA file, MGF files, and so on. And there are again icons here. I can try to look at the data. So if I click on this, the central pane shows me which chromosome, what's the format of that particular file, and that's true for your FASTQ file as well, and so on and so forth, right, BAM file. So one can look at that. On the left side here are tools available. So you can also search tools. For example, if I were to look at search GUI, it shows me that there is this tool called search GUI and then associated peptide checker tool. But again, for the sake of workshop, If I go, so you can actually spend a lot of time looking at this, and then if you go to the shared data, you can go to workflows now. And what we're trying to do is convert this FASTQ file in a protein FASTA file. So go to a workflow, and I'm going to select the first workflow, which is, or the second one here, which is workflow 1 RNA-seq database generation, right? So I go here and I say import. And once I import, I can say start using this workflow, right? And there's one feature which I didn't talk about, but this is the workflow now. So if you look here, if I click here, this workflow is in your, you've kind of transferred the workflow in your domain. You can modify the workflow. So I can show you that. So if you have this, right, I go to this, but so there are many things here, but I would strongly encourage you to go and explore these. So I go to this place called edit, and then hopefully very soon it will open the workflow output that we are seeing earlier. So you can see here, this basically shows the structure, and I think there's a way of reducing this, though I don't know how to do this on this computer. But you can reduce the size and look at this. But here you can actually see that there's a FASTQ file, the GTF file as an input, and there are these various tools that we talked about, right? So now, for example, if I wanted to go into this tool, right, and if you, for example, wanted to not have, I mean, I'm just giving an example. Let's say this says run individually, but if I want to say no, I want to change that to merge, I can do that and save this, but I might name this differently. Or I can change anything here and save it. And so you can modify these. You can also add things to these workflows. You can add inputs or outputs from these workflows, add tools to these workflows. So it's quite flexible that way, right? And then, so I'm not going to save this because, or maybe I will just have this, yeah. So, and from here I can now run the workflow, right? So maybe I'll save this and I'll run the workflow. You can also run it from the earlier place that I showed you. And when I'm running the workflow now, all I need to ensure is that I select the right input. So here, this says the first one should be FASTQ, but it's showing FASTA file. So I might go to, select the right one, which is the FASTQ file. Here, I need to select GTF file, so I select that. And thirdly, this is right. So this is the protein FASTA file that I have. And then I go ahead and I just click, and so each one of these, as you can see, are tools in that workflow, right? And there are 20 such tools. So, there are 22 such tools, right? And then I go ahead and just say run workflow. And then what will start happening is, and you should be able to see this very soon, is that the outputs, as you can see, have started piling up. So our initial inputs were here, and the first file has started getting converted from the first tool, and this will start happening for rest of them, and all these proteins or all these inputs will start getting generated. So this is how you are generating history, right? And maybe they just wanted to have a more dramatic way of saying I generated history, but I don't know. I mean, that's why it's called history. So at the end of it, we should have these outputs that are coming out, right? So I'm going to kind of just jump and skip to the next history, which is, so let's just imagine that, that history is run, right? Which it should in 12 minutes, we have checked that. So I'm going to go to history 2 and import that history. And sometimes I actually put in dates so that I know, which of these workflows were run on which dates. But in this case, I'm just saying Mumbai because I know if I just store the same thing, it might conflict with my, not conflict, but I might get confused later if I have to select it. What's that?
History 2 is the workflow 2.
History 2 is basically, no, history 2 is an output from the first workflow. So the ones that were gray right now, once you run them, you'll have history 2. So I'm kind of doing like a cooking show, right? I mean, just imagine this is cooked now. Let's look at the second one. Yeah.
History 1 will be the raw files then.
This will be the input files, yes.
Input files.
Yeah.
History 2 is the output from the
first workflow, and the third one would be from the second workflow. History 1 should be the result of our first workflow. Okay. So I'm just going to import it just the way it is. So that would have everything that is run, and I'll show you what we do next. I wanted to kind of just mention that if you have a completed history, you can convert this into a workflow. So I know this is a little advanced, but I'll just show you. So for example, this is your, after running the first workflow, and you generally do this in a stepwise manner, right? You can go to this wheel and then say extract workflow. And so now what happens is, if I have run a history, right, I can call it something, right, right, and then I say create workflow, and it will generate a workflow. Now I can share this workflow with you. You can take your input files and run on it. I can share the workflow with anybody. So the ability to share, so let's say if we are on the same Galaxy instance, I should be able to take this workflow, right, and then I can share it. So all I'll have to do is generate a link and then send this to you by email. If you click on this, you log in on this, you should be able to open that workflow. So that kind of helps you to not generate, use the same parameters. So coming back to that, so again, once you do this, what I'll do is maybe just show you. So we did the same thing now. We search the database and then we generate outputs. What I'll show you is the last output that is generated, which is basically everything together, right? You have input files, output from the first workflow, second workflow, and third workflow, which is one here. And then we can very quickly see if we can look at some of the features. And Galaxy has been in use in genomic studies for quite a few years now. We're basically just adapting it and using the ability that the fact that it's really strong in genomics and transcriptomics, to use it for proteogenomics, because then it's easier to merge these. So you can basically go through this really step-by-step manner. It also gives you information of basics of Galaxy, what are histories and all that. So it really is a very easy way of learning Galaxy if you're interested. So those who are interested, please go and use this. And then later, once you get a good feel for it, you can also contact us at galaxyp.org, I think, which is at the end. We have contact. So you can contact us galaxyp.org and we can suggest. So there are some sites in Europe right now which have got lot more infrastructure, so you can run your data sets on that, or you could also contact some researchers who are locally running Galaxy as well to run some of your data sets there. So this is the final summary, or the after running all the 3 workflows. As you can see here, it generates this output with the peptide, the number of spectra that it was identified with, what was the localization, which chromosome, what was the start site. It also gives you this link that you can use to open in a UCSC browser to look at, which is kind of an alternative way of looking at than IGV browser. So this is the MZ2 SQLite MVP output that I talked about. And if I click on this visualize an MVP application, it opens this application. No, so these ones here are not novel. These are all the peptide sequences from your MZ identimal file. But in your history, you have a place where it says novel peptides. So this 58 history is a list of novel peptides. So these are only, since this was a small data set, there are 8 of them here, right? And then the MVP application basically helps you to load that from Galaxy. And then here you would see only those 8 peptides. If I click on it, it opens this, and then so it gives you this peptide here, and now I can select whatever internal ions. Yeah, but it's not, so it's not been annotated right now. So if I add this, and you're right, I mean, if you think it's not a, if I start doing this, you might agree with it, but it's a very subjective thing, right? I might think, okay, 3 ions continuous is good, but you might not, and at least it gives you an ability to look into that. But yes, I mean, it helps you to do that, but you can also do it for other spectra and so on and so forth. So right now, oh yeah, so and then, so if I go from load from galaxy, if I do that, I should be able to, I can get those 8 now. And I can also go to view in protein, which I might not cover right now. I can then select this peptide. Because we went from a RNA-seq data, so it's from your protein FASTA file. But then it gives you ability to look at, yeah, so this is a genome-centric view. And now if I go here, so there are ways that you can look into this. You can expand this and look at the 3-frame translation of that particular peptide. You can see that peptide is here, the 3rd frame, and so anyway, I mean, again, this is for one peptide, but if you had 200 peptides or many peptides, you should be able to see that. So I think with that, I will end this.
In today's lecture, I hope you have learned there are 2 output files from RNA-seq to FASTA database creation workflow, which means the FASTA file and genome mapping files. You also got a glimpse of how one can search for novel variants using 3 basic steps shown by Dr. Pratik Jagtap. We also learned that how one can use Galaxy output files and use it in different online softwares like Integrated Genomics Viewer IGV to understand the mutations in a gene. In the next lecture, you will listen another speaker, Dr. Ratna Thangadu, who will talk about large scale data science. Thank you.